actually going to be smaller. If this cube is 5 centimeters by 5 centimeters by 5 centimeters, its total surface area is 5 times 5. That's the area of one of the sides of the cube. So 5 times 5, and then it's times 6 because you have 6 sides to the cube. So the surface area is 5 times 5 times 6, which is 150. And the surface area, or excuse me, the volume is 5 times 5 times 5. That's length times width times height. So the volume is 125. So the surface area to volume ratio is 150 divided by 125, which is 1.2. So as you can see, the larger a cell gets, the smaller the surface area to volume ratio. And this is going to put a strain on the plasma membrane. So it turns out that organisms are not just made of a bunch of big cells. Instead, they're made of trillions of little tiny cells, so they can maintain that large surface area to volume ratio. So as cells increase in volume, the relative amount of surface area to that volume, the ratio, is going to decrease, and that's going to put a strain on the plasma membrane. So this is going to limit your cell size, and your cells are going to stay relatively small. We're going to do this experiment in class. It's going to be a different color because we're doing it with something known as bromothymol blue, but you're going to be able to see cell transport in action. You're going to be able to visualize the fact that small cells can diffuse, or items can diffuse in and out of those cells faster than they can in large cells. It is important that you can calculate the surface area of cubes and spheres because you may be presented with a problem where you need to calculate these things. The surface area of a sphere is 6 times the area squared, and the surface area of a sphere is 4 pi radius squared, so 4 pi r squared. You're more likely to need to calculate the sphere surface area and volume. We'll do some of these in class. And then the volume of a cube is area to the third power, so area cubed. And the volume of a sphere is 4/3 pi r cubed. And don't forget that the radius is just the diameter divided by 2. So this would be the radius right here. Again, we'll practice those in class. There are 5 main components of the plasma membrane. So the plasma membrane is selectively permeable, meaning it lets some things pass through but not all things. And in order to accomplish this, it has 5 components. It has phospholipids, proteins that are embedded in the plasma membrane, cholesterol, glycoproteins, and glycolipids. First, let's focus on phospholipids. Remember, they have this phosphate head that is hydrophilic, meaning it's oriented or attracted towards water. And then you have these fatty acid tails, which are hydrophobic, and basically they don't like being around water. And so if you were to pour a bunch of phospholipids into an aqueous or a watery solution, they will spontaneously arrange as a bilayer so as to protect, so to speak, these lipid tails from touching water because they repel water. It is important to note that the structure of the phospholipid is largely dependent upon the fact that you have saturated fatty acids and unsaturated fatty acids in phospholipids. Remember, saturated fatty acids have only single bonds between the carbons that make up the lipid, but unsaturated fatty acids have double, or at least one double bond between some of the carbons. Double bonds cause the fatty acids to be kinked, and having only single bonds or being saturated cause the fatty acids to be straight. Your straight fatty acids, or your saturated ones, can pack together very, 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 very tightly. And so having a plasma membrane that's mostly composed of saturated fatty acids makes the plasma membrane very stiff. And having lots of unsaturated fatty acids means that they can't pack together as tightly. Think of it like crumpled paper. You can fit more paper in a box if it's nice and flat and you can just stack it up. But if it's all balled up, you can't fit as much paper in the box. And so your unsaturated fatty acids are kinked, therefore they don't pack together as tightly, and they cause the plasma membrane to be a little bit more fluid. So the percent of the unsaturated fatty acid keeps the plasma membrane more fluid. Another way of saying that is less viscous. Honey is viscous, it's like thick and can't move very fast, it's more stiff. And having unsaturated fatty acids helps to maintain that fluidity of the plasma membrane. Cholesterol is also going to contribute to membrane fluidity. Cholesterol can actually help make the membrane more fluid or less fluid depending on the temperature. So at high temperatures, the plasma membrane is in danger of basically being too fluid, falling apart. And at high temperatures, cholesterol prevents the plasma membrane from melting, essentially. It's going to raise its melting point so it doesn't fall apart. At low temperatures, cholesterol prevents your phospholipids from clustering together or lumping together and stiffening. So cholesterol keeps the plasma membrane from becoming too stiff when it's cold. So it's almost like a buffer. It keeps it from being too fluid or not fluid enough. So this is going to be, or this is just another diagram showing you how phospholipids self-arrange to a phospholipid bilayer. It is important to remember that nonpolar substances, if they're very small, are going to be able to squeeze through the phospholipids. However, a polar substance, no matter how small it is, cannot squeeze through the phospholipids because the lipid bilayer is going to repel them. Because remember, they're nonpolar, so a polar substance won't be able to squeeze through. Obviously, if something's really large and nonpolar, it's also not going to fit through, just like a fly can't squeeze through the screen on a window. So you're going to need some substances that are polar and large to get into a cell, so they're going to have to get through in a different manner. So first, let's discuss what can diffuse through the phospholipid bilayer and what cannot diffuse through the phospholipid bilayer and has to go in and out in a different manner. So anything that's very, very small and uncharged or nonpolar is able to go directly through the phospholipid bilayer. Again, it has to be small and nonpolar. This includes fats and other lipids and very small uncharged particles like nitrogen gas. Nitrogen gas and oxygen gas can go straight through the phospholipid bilayer because they're nonpolar and they're very small. Polar molecules like water cannot go directly through the phospholipid bilayer. They're going to need to go through a protein channel. Ions and other charged substances also need to go through a protein channel. Again, they can't interact with the nonpolar fatty acid tails of the phospholipids. Also, large molecules, even if they're nonpolar, are going to need to go through a protein channel because they're too big to go in and out of the cell. They can't squeeze through the phospholipid bilayer, just like the fly can't squeeze through the screen door. Your proteins are going to contribute to permeability of these larger molecules. Basically, your proteins are going to enable larger molecules and polar molecules to enter and leave the cell. We say your plasma membrane is semi-permeable because it controls what goes in and out. It lets some things in, but not others. And proteins are going to let in some items, but not others. So you have specific proteins that let in water, and a specific protein that might let in salt, and a specific protein that might let in sugar. So they're specific as to what they enable to enter and leave the cell. Now don't forget that the inside of the cell and the outside of the cell may contain water, and that is again why your fatty acid tails associate or arrange with one another so that the lipid tails are protected, so to speak, from the aqueous environment inside and outside of the cell. So proteins are the perfect molecule to build your plasma membrane because they're composed of amino acids. Don't forget that amino acids have those R chains. Remember, an R is just some sort of collection of carbons. And so those R chains might be polar or nonpolar, which means that your proteins can have polar sides to them and nonpolar sides to them. So you might have a protein that has a hydrophilic region and a hydrophobic region, so it can arrange appropriately in the plasma membrane to enable certain things to enter and leave. Let's take a closer look at that. So this would be a membrane protein right here. The purple portion would be polar because it's associating with the aqueous environment inside and outside of the cell. So again, the purple would be polar regions. The kind of magenta or reddish side of the protein would be the nonpolar regions because again, they're interacting with these nonpolar or hydrophobic fatty acid tails. Now, if this is a protein channel that enables polar items to enter and leave the cell, the lining of the tunnel would also need to be polar. Here are two specific examples of membrane proteins. The first one is an aquaporin. It is exactly what it sounds like a pore that lets water go in and out of a particular cell. So bacteria have these. It's a water channel. And so again, the part of the protein that's right next to the fatty acids is going to be nonpolar. The part that interacts with the inside of the cell and the outside of the cell is going to be polar. And then the lining of the channel, like almost like the tube of the protein, the lining of that tube also needs to be polar so that water can associate with it and go in and out. This is very similar. This is a protein pump channel. Photosynthetic bacteria, also known as cyanobacteria, have this. And protons are charged, and therefore they cannot, even though they're small, they cannot go directly past the fatty acid tails of the phospholipids. And so similarly, they're going to need to go through a protein channel. So not all membrane proteins function with transport. Some can act as enzymes. Some can act as receptors and be part of cell communication. You have other proteins known as antigens that help to identify a cell. It identifies a cell as a skin cell or an eye cell, or really identifies it as a cell that belongs in your body and not a pathogen. And this is important for your immune system. You have other proteins that help your cells to attach to one another so they can connect the plasma membrane of one cell to another cell. And then you have other proteins that can help connect the plasma membrane to the cytoskeleton itself, so connect itself to parts inside of the cell. So let's look at some of these different membrane proteins. First off, we have two classifications of membrane proteins. We have peripheral proteins and we have integral proteins. A peripheral protein is going to be on the surface of the plasma membrane. It will not actually go through the plasma membrane. So you've heard of your peripheral vision before. It's just like the vision that you have on the side of your head. And so peripheral proteins are also just going to be on the side of the plasma membrane. So they won't actually go through the plasma membrane. You don't specifically have to know this, but one example of a peripheral protein are your proteins that help identify your cells as self and not as foreign invaders. So your antigens are peripheral proteins. But again, you don't really have to know that specifically. Your integral proteins are proteins that penetrate the lipid bilayer, and they actually go across the whole plasma membrane. An example of this would be a protein channel that enables things to go in and out of the cell. We call our plasma membrane the fluid mosaic model because substances can actually move across the membrane. They're not stuck where they are. So you actually can have proteins that can slide around in the plasma membrane, and we say it's a mosaic because it's not just made of simply one thing. Let's take a closer look at the carbohydrates in your plasma membrane. Again, like I mentioned previously, they can function with cell recognition. Your antigens are proteins that contain specific carbohydrates attached to themselves. An example of this is your major histocompatibility complex, or MHC. This is very important for your immune system because it helps your cells identify which cells belong in your body and which cells do not belong in your body. So this is going to be the basis for your immune system. We will learn much more about your MHC later. Now let's look at our cytoskeleton. Your cytoskeleton is not part of the plasma membrane, but it is the network of fibers that goes throughout your cytoplasm. Your cytoplasm is not just, the inside of your cell is not just goo. It has this network of fiber that crisscrosses within the cell to help maintain the shape of the cell, to help anchor organelles, to help the cell move around, and basically to help all of the different organelles work together. You have three main components of your cytoskeleton. There are microfilaments, intermediate filaments, and microtubules. They are all just different types of protein fibers. Your cytoskeleton actually helps to comprise cilia and flagella. Remember, cilia kind of look like little hairs, and flagella look like little tails, but they're actually comprised mostly of cytoskeleton. And this is just one more example of your cytoskeleton. You can see here, this is your mitochondrion. Every cell is going to contain probably multiple mitochondria, but this mitochondrion is anchored to the cytoskeleton so that it doesn't just move all over the cell, and it can associate with different parts of the cell. And so this is a microscopic image of a cell with fluorescent dye that's tagged different parts of the cytoskeleton. So actin and microtubules are two types of the cytoskeleton. The actin was dyed red, and the microtubules were dyed green, and then the nucleus was dyed blue. So if you look at this cell, you might say, why does the cell have two nuclei? And the reason being is this cell is undergoing cell division. So the nucleus has already split, and now the cytoplasm is about to split. And your cytoskeleton is involved in that as well. So if you remember from learning about cell division, your chromosomes will line up in the middle of your cell, and then these little strings or microtubules are going to yank your chromosomes apart. Those are microtubules, and so they're involved in the process of cell division. So I'm getting a little bit ahead of myself. We'll learn more about cell division later. So I hope that was helpful.